Okay, in this video, I'm going to be explaining some cancer mechanisms of how they survive, and also some foods that can help. So the first one is salmon. Salmon has astaxanthin, and the first mechanism that a cancer cell survives is with C-flip and FAD, FADD. So basically, C-flip inhibits death receptor-mediated apoptosis by competing with caspase-8 at the FAD complex. But astaxanthin has been reported to downregulate C-flip in certain cancer cells, which makes them more susceptible to facile apoptosis. Astaxanthin promotes FAD-mediated recruitment of the caspase-8, enhancing extrinsic apoptosis. Now, there are different caspases. So caspase-8 is just one of them. There's also a caspase-3 and a caspase-9. So basically, astaxanthin in salmon can sensitize cancer cells to death receptor pathways by reducing anti-apoptotic C-flip levels and supporting FAD function. So basically, the C-flip that cancers have is anti-apoptosis. So cancer cells use C-flip as a way of escaping programmed cell death, but astaxanthin can stop that. So there's two other ones. There's BAX, which is pro-apoptotic. Basically, that's anti-programmed cell death. And then there's a BCL2, which is anti-apoptotic, which means preventing apoptosis or programmed cell death. Now, what astaxanthin happens to do is it upregulates BAX, promoting apoptosis, and downregulates BCL2, stopping of apoptosis. So, by reducing survivin levels, this facilitates caspase activation. Now, astaxanthin also affects this next one, which is CD80, CD86 antigen-presenting cells that bind CD28 on T cells, enhancing T cell activation, and therefore giving cancer T cells to fight against. So CD80, 86 on dendritic cells, which is a form of an antigen-presenting cell, by upregulating the CD80, CD86, this improves T cell priming and anti-tumor immune response because that the T cell is activated by the antigen-presenting cell by binding onto the CD28 on the cell. The next mechanism, next cancer escape mechanism, which astaxanthin happens to also influence or alter, is PD-L1 on tumor cells, binds to PD-1 on T cells, which equals immune suppression. Astaxanthin happens to downregulate the PD-L1 expression in some cancers, reduces PD-1-mediated T cell inhibition. So basically, astaxanthin takes away the PD-L1 on the cancer cell, and astaxanthin also takes away the PD-1 on the T cell. So therefore, the cancer cell can't bind to the T cell. The T cell is free to attack the cancer cell, and the cancer cell has no defense mechanism, or at least in the case of PD-L1 and PD-1. The mechanism that cancer cells thrive and survive on is TNT, which is short for tunneling nanotubes. And basically, through these tubes, cancer cells, even from long distances, they connect with other cells, either healthy cells, or they even communicate with cancer cells. So in the case of healthy cells, they transport like damaged mitochondria, damaged DNA, misfolded proteins over to healthy cells to make them unhealthy and possibly even cancerous. Curcumin from turmeric is known to reduce TNT formation by modifying actin polymerization, which is beyond the scope of this video. I could explain that in a different video. Curcumin also disrupts actin-based nanotubes, disrupting the RHO GTPase activity and RAC1 and CDC42, which is what cancer uses to form the tubes themselves. So TNTs also have this other name called philopodia, which are actually protrusions, explaining how the cancer cells connect to other to other cells, whether if they're cancer cells or other healthy cells. So curcumin also lowers reactive oxygen species. Cancer cells, they use that to trigger TNT growth. So antioxidant deficiency, such as vitamin C and vitamin E, can actually promote tunneling nanotube growth. So it's important to get your antioxidants in. Curcumin also limits stress-induced TNT formation. Cancer cells, they also have a mitochondrial rescue transfer, which I'll have to cover in a different video. And they do that through the TNTs. So a cancer cell connects to another cancer cell with a TNT, and then they do this mitochondrial rescue transfer. Because cancer cells, they transfer mitochondria through those tubes. Curcumin also disrupts mitochondrial quality control, which means that cancer cells cannot keep sending other cancer cells mitochondria as a survival mechanism or to save their other cancer cells. So basically, cancer cells can't rescue each other thanks to a curcumin. Curcumin also weakens the communication signals that drive TNT growth, such as STAT3, also hypoxia. Another fruit that can lower STAT3 so that the tunneling nanotubes are reduced is a papaya. So papaya has many other huge benefits besides just lowering STAT3. Okay, hypoxia. Basically, that happens because of iron deficiency, because the red blood cells are not getting iron. Red blood cells need iron to deliver oxygen around the body. And if red blood cells do not have that iron, then that's going to lead to hypoxia. And tunneling nanotubes they grow from hypoxia. So a good way to avoid iron deficiency is to make sure that your zinc and iron is not, that your zinc and copper levels are just right. Zinc is not too high, because what that's going to do is that if zinc is too high, that's going to reduce the copper level, and then iron is not going to work because proper copper is needed for iron metabolism and transport. And if there's not enough copper, then there's going to be hypoxia because red blood cells are not getting the iron. Also, a good way to reduce the zinc, if your zinc is too high, is with quercetin. Quercetin binds zinc, and so that you can reduce it. Therefore, your copper levels are going to be just right for the iron to be able to work. Curcumin also enhances immune recognition when TNT activity decreases, because cancers use TNTs to share PD-L1 vesicles with other cancers. What that means is that the other cancer cells are going to they're going to get that PD-L1, and then they are going to start attacking T cells because the T cell has the PD1 or the PDL. So if cancer cells are attaching to other cancer cells with TNTs, they are through the tunnels. They are transferring PD-L1s to other cancer cells, which makes the other cancer cells lethal and more likely to attack your T cells. So that's the end of the video. Like, subscribe, leave your insights in the comments, and I'll see you in the next video.